all linked to open source data sets, evaluations, models, and so on that are available from our net website. So you are very much encouraged to check this out. So the use of AI in science and scientific discovery and medicine is not new, and we are seeing in the last several years how AI has beginning to have a real impact on fundamental new scientific discovery, as well as how scientists go about their everyday research activities. We're seeing generative AI changing the way we do science. And even more importantly, and perhaps more exciting, is also to see how AI is increasingly used to augment research and provide insights that might not have been possible just a few years ago. What does that mean? Here's an illustration of that. Just a few weeks ago, AI-enabled drug discovery, which is an area of scientific discovery that is concerned with developing AI models that can help us design and discover new drugs, has reached clinical milestone. And by that, what I mean is that for the first time it happened that a drug, a chemical compound, which can be thought as a molecular graph, was generated by generative AI models. It was designed such that it binds to a biological target, which was also nominated by multimodal AI models. And that combination of a drug candidate and a target went way beyond academic in silico benchmarks to clinical trials. And now 2 weeks ago, there was a phase 2A clinical trial of that candidate drug completed successfully, showing an acceptable safety profile and positive sign of efficacy for this candidate drug to be effective against idiopathic pulmonary fibrosis, which is a disease for which no good treatments exist yet. So this is very exciting. It's a proof of concept how AI can help science and medicine in a way that was just not possible a few years ago. So AI and machine learning, however, are not new in biology, are not new in medicine. For the last several decades, we as a field have been training and developing various kinds of machine learning models and applying them to various kinds of problems in biology and medicine. So what is different this time? Why there is so much enthusiasm in the use of AI across science and biology? So the short answer to this big question is the kind of new capabilities that AI has offered in the last several years, which broadly we can think of under the umbrella term of foundation models. The big idea of foundation models is that we'll collect a single massive consistent machine learning data set, and then we will train a model based on that data set, and we'll do that without requiring that the data set is fully labeled through manually sourced annotations. The number of foundation models go out there, including in the general domain, ranging from GPT, recent reasoning models, DeepSeek O1, O3, as well as open source models such as Llama family of models, as well as increasingly models developed for areas of biology, such as protein language models, ESM, structure prediction models, the alpha family, as well as genomic foundation models. What drives the success of foundation models are broadly three things. A, the availability of large-scale data sets, particularly into areas of biology, structural biology, and single-cell biology, where we have seen the most activity in foundation models in recent years. Second, hardware, and these are large-scale GPU clusters we can train these models on. And third criteria are algorithmic advances, specifically self-supervised learning and transformer architectures. So in my group, we are working under this umbrella with the overall arching vision or goal to create AI scientist systems. And AI scientists for us are models that are able of skeptical learning, skeptical reasoning that can leverage multi-model, multi-scale data, and do that in connection with experimental validation of AI outputs with the goal and vision that eventually these models and these systems will be able to operate on its own and act independently of human scientists or collaborate with them to go after the toughest challenges. So under the AI scientist paradigm, we're operating on two big areas, where one is concerned with developing models that can help us understand what are the rules and functions of molecules, how to translate those functional insights into potential drug targets, and design chemical compounds across those targets to help with discovery of and design of drugs from scratch. And the second big area of research for us is to understand existing medicines and existing kinds of interventions and treatments and maximize their use across many diseases that have for which we have limited understanding of what they do and how to treat them well. And so in the rest of today's talk, I want to share with you a few vignettes of the AI scientist paradigm by sharing models that we have developed that operate across these 4 scales of biology. And I'll start with the far right, which is that scale which is the human scale, where we're developing models that can take the clinical and multimodal data for a patient and then match those patients again to treatments that are most likely to be effective and safe for a patient. And then we'll go down to the cellular level, the cellular understanding, how cells and cellular environments are organized in human body. Within those cells, we have many, many biological networks or molecular networks where molecules of different kinds interact with each other. So we'll look at modeling of interactions, and if there's time left, also we'll discuss some of the recent work that several mentors or organizers here in this summer school have developed on how to understand the functions of molecules much more broadly. So let's start with this part of the far right, which is the therapeutic modeling and how we can go about very precise matching of patients towards therapeutic effects. So the motivation for this work is, of course, large language models that have been with us now for several years, with the goal of capturing really broad amounts of scientific literature, very diverse data sources that integrate clinical insights with molecular insights, and that we can then prompt in various different ways to ask questions and see answers to those questions, and those questions can be therapeutic questions. What we have been increasingly interested in is to go one level up, which is from training these large language models to next token prediction, to think about using these type of models in the context of other kinds of biological and biomedical data, where large language models can use tools, and those tools can be other machine learning models. They can send out query requests to external databases in real time, ask for the data they need to solve the problem that the human scientists or clinical researchers would ask them to solve. And so I will show to you and walk you through the next couple of slides what is the paradigm of AI agents, and I will illustrate that through one agent system that we have recently developed. So when I say AI agents, we really are not talking about these kinds of agents. I hope you're not too young for not knowing this movie, and so I don't know about that. So we'll talk about these type of AI systems that are agents that can perform certain actions in the real world. So the agent I will walk you through is Text Agent. It's an open source model that comes with the data sets, evaluations, open model weights, training and inference code. It's on this website. So it's a work that was led by 2 fantastic postdocs in the group, Shanghua and Chengkun. They have developed Text Agent that is a reasoning agent model that can use over 200, now almost 300 tools that allow this agent to reason and across all drugs developed since 1939, do that in the context of tens of thousands of disease phenotypes that are phenotyped and described in monarch initiatives, and also retrieve clinically validated insights to ground those models from open targets. So when we start unpacking a bit, well, what is Text Agent and how it's organized, so we can think of Text Agent as a system that the user will interact with through natural language interface, so you could read from left to right. On the left, human scientists would provide information that they're interested in, of a task that they're interested in solving, and that might consist of some vignette of a patient that is experiencing a certain health situation, information about the disease that the patient might have or suspected to have, and some idea about, well, what could be a potential drug or the mechanism of action that the drug should have in order for the drug to be effective for this patient. And so this input prompt will then be translated by an LLM into a TX agent system, where TX agent would then realize that there might be tools or data sets out there that it could leverage in order to answer this complex question. And finally, at the end, what it will produce is an answer to this task or question that was asked to solve, justification, and a reasoning trace, which consists of a set of steps that the agent performed in order to solve the task. There are three key features that TX agent has. So first, TX agent is able of open-ended reasoning. And practically what that means is that you can ask it any question, including questions that the agent was not explicitly trained on, and it will produce answer as free-form text output. So it's not anymore about closed-form prediction, where the output of an agent or an output of an LLM would be a set of predefined labels or answers that the agent selects from. The agent is capable of multi-step reasoning, which means that it can solve more complex therapeutic questions that require decomposing the tasks into subtasks that need to be solved, and then compiling the solution of those subtasks into the big task. And finally, I said that this is a reasoning agent, and so we will see how that reasoning agent compares to state-of-the-art reasoning models such as HARDA. Here's a very, very simple example of a query to TX agent, and I'll also show a demo later on that's a bit more involved. But in the very simplest case, you can imagine that we would ask, we would start from the very top where there's a question. So we might ask, what adverse reactions should be considered when taking Alpitrek? So that's a problem to consider. And so what TX agent would do, it would realize, well, for this question, what would be really good to have is some tool that I could query about adverse events for this drug. And then it looks into its toolbox through a mechanism called toolrec and realizes, well, there is an API at FDA that you can send requests to with drug name, and you can get back information about currently listed on the drug label adverse events of any drug. So it realizes that, and so what it will do, it will use this tool, send an API request out there, in real time get the data from FDA, and realize that, well, that data is actually useful for answering this question. So it's because it was a relatively simple question. So it gets drug label for Alpitrek, realizes the information that it has to extract from the drug label to answer the question is sufficient, and then it will produce an answer. And so this is just an illustration of how the model can extract and leverage external tools and not anymore rely solely on parametric memory that is in the memory of a large language model while it was trained on lots of literature. How is that possible to achieve? So the keynote here is what we call AI communication, where we don't think about now AI models as acting independently of each other, but instead they can talk and communicate to each other and exchange information. So I think a very useful analogy of that is really HTTP protocol, which has transformed how we view internet today. Before HTTP protocol, what we had, which was like many years ago, was just a set of disconnected web pages that were very hard to index or traverse. With HTTP protocol introduced, it was possible to start connecting pages to each other, link them to each other, and that's how internet emerged, and that led to all kinds of great things we have today. So an analog, a similar protocol, set of protocols are also emerging for agents, and one of the popular one is called MCP that was released by Anthropic. There is another one called A2A that stands for agent-to-agent communication that was released by Google just a few weeks ago. And what these type of protocols allow us to do is to start connecting text-based large language models with other kinds of machine learning models that can be based on geometry, they can be based on graph machine learning, they can be based on vision models, other kinds of tools such as external databases and APIs, and do that really effectively. And so that's how we can realize and create these agents that start using tools. Okay, so what are the tools that TX agent uses? TX agent currently uses the version that is out there, supports 211 tools. Those tools come from trusted resources, and TX agent uses them for real-time knowledge retrieval. there's a very interesting consequence of real-time knowledge retrieval, which means that the model does not get outdated. So for example, if there is a new drug approved today, and we trained the model 6 months ago, the model can still reason about this new drug as long as it can be information about it can be accessed through the same set of tools and APIs as they were developed for previous drugs, which is generally the case. So that allows the model to really remain current for longer periods of time than what was possible before. For each of the 211 tools, we have this very simple tool description that consists of information about what's the tool, how is the API called, parameterized, and its description in terms of inputs and outputs. And these tools can be specified really, really easily. While in DX Agent, we only consider API-based tools, Sean has developed a universe that consists of not only 211 tools across FDA, open targets, and Monarch initiative, but also brings in other models and other data sources. Here I'm just listing a few that range from knowledge bases, NVIDIA, NIMS, various models that our lab has developed, including geometric deep learning models, Pinnacle, processing foundation models, just Procyon, another agent system, Prism, and many other model context protocol services. So everyone can use the tool universe, and it's as simple as you can plug in your tool universe to Clot if you use the closed source models. And so when you have the input window in Clot, you can just see that there's a tool universe option and which tool you would like to use for tool universe to solve your tasks. But you can also use this tool universe to train ML, to train reasoning models. So how can we train DX Agent reasoning model? The issue here is that we're not just training a regular LLM. We know how to train a regular LLM for next token prediction or the self-supervised learning. When we are training a reasoning model, things become a bit more challenging. So the training data set has 3 key components. The first component is tool description. This component has 211 entries for 211 tools. For each tool, we have the mapping of the tool to the external representation of tools used by the agent. And that mapping practically is just a JSON object that contains information about the tool, its parameters, and documentation of the tool. The second component in training the DX agent is a large database of therapeutic questions. So we built a data set of 85,000 therapeutic questions that are questions about adverse events of drugs, that we have seen a few slides ago, indications, contraindications of drugs, combinations of drugs and what combinations might work well or not work well for patients with certain pre-existing conditions and so on. And the third component is a data set of reasoning traces. This is a crucial component because it allows us to teach the model when to use a specific tool. And so this TraceGen data set has 85,000 reasoning traces. Each reasoning trace is a sequence of actions and which are calls to different tools and results of those tools. And that information is used to teach the model which tools to use for what type of question. So these 3 data sets together are then used to train a reasoning model starting from either Llama or QN3, and then make a reasoning model out of it based on these 3 components. Once we have the model trained, well, how can we evaluate its reasoning capabilities? And well, how well does it perform? And how can we benchmark it if it can answer open-ended questions without those questions being pre-specified before? So we built a reasoning benchmark based called drug PC, which stands for drug prescribing cards. Shown on the left are examples of 3 drug prescribing cards. For every new drug approval, eventually FDA will release a document that might have 40 to 60 pages of content, and many sections in that document describe everything from the chemical structure of the compound that is a drug, to its pharmacokinetic, ADMET properties, adverse events, clinical trial results, Should the drug be used with elderly patients, or should it be used with patients that are children? Are there any contraindications of drug, and so on? And so across these different sections of the drug prescribing cards, we created benchmarks of them. And the goal is to see whether TX agent can effectively predict and generate content for a specific section of a drug prescribing card for drugs that were approved very recently beyond after the knowledge cutoff date of the model. So on this slide are shown results of the model of TX agent and alternative models across various sections of the prescribing card on this benchmark. What you can see on y-axis is accuracy, higher is better. On x-axis, we have different blocks of bars that show how well the model can perform when it is asked to answer questions about dosage, administration of the drug, or clinical information or clinical pharmacology, and so on and so on. And so what we compare TX agent model to, we compare it to the base reasoning model the TX agent is built off. So we can see here TX agent AP that was built with Llama, and its comparison to Llama model alone, and with 3 strong lift of performance, indicating that these new capabilities the model has go above and beyond what a direct LLM could provide you alone. We also compare TX agent to a larger model that does not use tools, such as here GPT-4o, as well as a few alternative models that can use tools as well, but cannot use a large number of tools with overlapping capabilities. And we see here strong performance when compared against larger LLMs as well as tool-use LLMs. We can ask TX agent personalized questions where, as you can see here on the left, is an example of a patient vignette where we have a 60-year-old female patient with a history of myocardial infarction, and the patient is working on beta blockers, have no renal or hepatic impairments, and the question is which medications would be most appropriate to add to the pharmacy of that patient to reduce cardiovascular risk. So this is like a complex question. This is not a question that can easily be mapped to a simple prediction of a link between a drug and a disease that might have, because it requires simultaneously to consider potential adverse events, which is renal, liver function, liver and kidney function impairment, other diseases the patient might have. And so both in closed form reasoning, where the model sees a set of candidate options like A, B, C, D, and needs to select the appropriate one, or where in open-ended reasoning, where the model has to generate answer from scratch, TX agent performs better than alternative models we compared the agent against. Then we were also interested in comparing this reasoning model to state-of-the-art reasoning models, if we are arguing that TX agent is a reasoning model. So we compared TX agent AP model to a DeepSeek family of models, and specifically 3 models in the DeepSeek family, including the AP70 being 671B model. And we were quite surprised that TX agent performed favorably when compared against these much larger reasoning models. How can we explain that? I'm just illustrating one explanation of this result here on the right part of the slide, where we can see a therapeutic question that both TX agent and DeepSeek were asked to solve. And then the way DeepSeek approached that is that it took that complex question in the input prompt and started decomposing it to simpler tasks and start initiating a sequence of reasoning steps to solve that complex tasks. But the problem was that somewhere early in the reasoning process, it made a mistake, and there was not able to effectively recover from that mistake. And because this reasoning process is sequential in nature, the errors compounded, and the model was not able to recover from that. That's in contrast with the TX agent, where we require that each step in the reasoning process is grounded in external knowledge that is defined by the use of tools and the data retrieved via tools, and that allows the model to hallucinate less and provide more accurate response. There are a number of other ablation and sensitivity analysis that we did in this project. For example, we were interested in question whether adding more tools is helpful. Is it helpful to have 211 tools, or could we do the same with just 5 tools or 10 tools? And so you can see here in panel D that when the number of tools in the tool universe is increased, that generally leads to better performance of the model. Then in the second row, what we see is performance of the model when it is allowed to generate as many tokens as it wants during the reasoning process and perform many reasoning steps, versus when the model is just forced to generate an answer immediately in a one pass. And generally we see in panel G, when the model can generate multiple thoughts and actions in response to the thoughts it generated, the accuracy of the model will go up. So that's all great and fantastic benchmarking of model performance. What we are interested in doing is also to see whether we can start using these models for new exciting medical research. So let me just share a few vignettes of that. So what we are doing currently is evaluating AI predictions generated by TX agent by emulating clinical trials and do that at scale. So we're working here with Claudet's Innovation, Bill Melinda Gates Foundation, and CZI to emulate clinical trials for 60 diseases. For each of those diseases, we have appropriately defined medical proxy endpoints, and then we leverage large EHR reference data sets to emulate clinical trials for each of those 60 diseases in the US and 13 other countries. Here's just an illustration of one such trial that was recently emulated by Ayush, another student in the group. So Ayush has used the model to predict and nominate drugs that might likely have protective effect for Alzheimer's disease. So one of those drugs that was nominated by him in the model that he used was dapagliflozin. And so now we have this prediction, this an AI prediction, we predict that dapagliflozin, which is a drug that was initially developed for diabetes. It's not used for Alzheimer's disease. It's not prescribed and given to patients with Alzheimer's disease. But the model says that it will protect patients against Alzheimer's disease. So the model says it might likely have good effect on Alzheimer's disease. How can we make sense out of this? So the approach is that we look at large EHR database, so database of electronic health records, and we emulate clinical trial, but we emulate clinical trial, and we do that so by creating 2 cohorts of patients in that EHR system. We'll call one cohort treatment cohort, the other will be control cohort. So treatment cohort are sets of patients that take this drug. They take the drug for diabetes because that's what the drug was initially indicated for, and they do not have Alzheimer's diagnosis. Then we have a control group, which is a group of patients that are age and sex matched as the treatment group. And those are patients that are not taking this drug, and they also do not have Alzheimer's disease. Great, so we have this treatment and control cohort, and then we will track patients for 10 years and see how many patients in the treatment group developed Alzheimer's or Alzheimer's-related disorders. How many patients in the control group developed Alzheimer's? And if there is substantial gap, significant gap between Alzheimer's-free survival in the treatment group versus control group, then that would suggest that this prediction that the AI model might really have protective effect when it's used with real human patients. So these are results of the actual trial that was emulated at Mass General Brigham on 194,000 patients. And we can see here in red is the control group, in green is the treatment group of patients that were put on dapagliflozin predicted by AI model. And we see on the y-axis probability of how likely patients will survive without being diagnosed with dementia, which was one of the proxies for Alzheimer's disease. And we can see that those that were in the treatment group, this trial would suggest, and we can calculate the hazard ratio and do some rigorous statistical analysis, they would be less likely to develop dementia, suggesting that, well, this AI prediction might be quite interesting to look at further. Here's another example of what we also are doing with AI predictions made by models that I've just described. So here we use a related model to the agent called Cypher. This was also the work done by Ayush. And Cypher was used to nominate drugs that might likely be beneficial for patients with bipolar disorder. So the way we tested that drug was that we then worked with our experimental collaborators in the Church Lab and the biogenetics department, and they had data from patients diagnosed with bipolar disorder. And for those patients, they were able to extract blood samples. Based on blood samples, grown brain organoids from patients with bipolar disorder. So shown here on the left, in the bottom, is an image of how stained brain organoid derived from patients diagnosed with bipolar actually looks like. So what we have done then next is we have now a collection of brain organoids, and to those brain organoids we administer this predicted drug, the dosage that the model predicted. And we check, well, what happens to those brain organoids? Do they look more like healthy brains, or nothing really happens much? So what you can see here on the right are results of that experimental validation. And these results are gene expression values of this relevant cell types for brain organoids. We also have other omics data generated, but I'm just showing here a very simple gene expression analysis of those organoids that were treated with the drug predicted by the model. I want to draw your attention to these three groups that are highlighted on the right part. So what you can see, control group is a set of organoids from healthy individuals that are not that do not have BP. In green group, we have for every column in green group is a brain organoid of patient derived from patient with bipolar disorder that was treated with the drug predicted by our AI model. And so you can see that this blue-red pattern, which are differential gene expression analysis on the relevant for the relevant genes, is very similar to the control group, which is good and strongly encouraging result. And what you see in the red group are expression levels of organoids derived from patients with bipolar that were not treated with the drug predicted by a model. And you see that the pattern of genes that are activated is quite different from those organoids that were treated with the drug predicted by the model. So these are two illustrations of how you can, as computational scientists, you might be computer scientists, mathematicians, electrical engineers, applied mathematicians, and on. You can have an impact on medicine, on scientific discovery. And I think they're just like so fantastic and so exciting in these days. Okay, so we have these models are all open source models. They come with open data sets, evaluations. We are also running a competition at Durinx 2025 called CureBench, for which we'll give out 100K worth of awards for to those who will make the strongest submission and will win the competition. The website is already up. The formal announcement will go out in the next few days. But if you go to CureBench AI, you can start familiarizing yourself with different tracks that we defined for evaluating reasoning abilities of today's AI models for drug decision making and therapeutics across scale. Okay, so what I have here is a very quick demo. So DX agent is this like simple agent that allows you to ask questions. And the way it actually runs, it's like that. There's a question. This was a question of a 70-year-old woman that should be prescribed drug, yes or no. And the model will not just immediately answer the question. It will realize that there are external tools in the tool rack that the model can leverage effectively to get information about this drug from FDA. So to do that, get the data about the first events, warnings of the drug that are in the box of the drug, and then leverage, bring that all that information in its own parametric memory of an LLM, reason about it, and then finally produce an answer. What we also have now, it's not yet public, but I'm super excited, it's a DX agent matrix. And this name was invented by Shanghua, not myself, which I'm usually one who has crazy ideas for names. So as you know, what we have in UV matrix, we have lots of different agents. So TX agent matrix is actually a version of TX agent that can automatically spawn other agents. And what this can be useful for is to allow the agent not only to perform strong retrieval tasks and help with asking questions about drugs that already exist, but can go beyond the boundary of the knowledge that exists in a single LLM. So I'm not sure we'll go to this. Maybe this will take like 2-3 minutes. But then we start with a question, and then the agent now will say, well, this is an interesting question. I could generate a few different research plans to execute that could be informative for this question. And so what it will do then is for each research plan, it will initiate its own TX agent instance and will say, okay, you should go and execute the first research plan. You should go, another agent, TX agent will go and execute the second research plan and the third one and so on. And then this community of TX agents will then execute different research plans and send all the results back to the master's TX agent, who will then interpret the results and produce the final answer. So this allows us then to also generate novel scientific hypotheses, not only hypotheses, but generate research plans. It's like a sequence of steps that should be performed to arrive at a specific conclusion. And so, as you can imagine, because now we have multi-agent system, not just a single one, this entire pipeline might also take a few minutes to execute. And kind of that's what's happening here. Okay, I guess I have 8 more minutes. Okay. Oh, we started late. Yes, absolutely. So great. So we'll switch the topic a bit, but I saw that there's a question there, so happy to take one.
What's the difference between those multiple agents?
Those multiple agents are, once they're trained, they're prompted in slightly different ways, and they use different sets of toolboxes. So one agent uses clinically oriented toolbox. Another agent uses toolbox that allows the agent to interact with models trained on molecular or omics, non-patient data. And this allows us then to think about the therapeutic hypothesis based on molecular, multi-omics, structural data, as well as on the other side, clinical evidence, genetics, clinical trial results by different agents. Okay, yes, another question.
You mentioned that your agent uses trusted sources to help brand it, but like, are you considering if that those trusted data sources contain conflicting information? Like, are you handling that, or are you just implicitly dealing with it?
Great question. At this time, we are assuming that those trusted sources, which are trusted sources. So we do not have any notion of any notion of probabilistic interpretation of the knowledge retrieved from the trusted resources. Human user scientists can decide what are the sources that it would trust. But then, for example, it would assume, well, if it comes from open targets, if it comes from FDA, I will assume that that knowledge is to be trusted. That generally is the case. It helps with reducing hallucinations substantially over using LLMs alone. But you're right, we do not have any notion of conformal estimates put on the model. That's sad. We have really exciting project on this topic led by Inte and Ying, two postdocs in the group who will have a paper out hopefully in 2-3 weeks on a model that is called STRAT-ZP, which can be used with any medical foundation models for conformal prediction. But the TX agent does not have that. Yes.
I have a follow-up question on the
Yes.
same type. So we spoke about hallucinations.
Yes.
And we know that all elements hallucinate to some extent.
Yes.
So also another flip side is a drug which can have one set of side effects, might not have another set of side effects on another patient. Right.
Correct.
So how do you know that the prescriptions that the LLM agent provides are like robust enough, or how can we reach them?
Yes, you're absolutely right that the drug that might be used for one patient might not have the same adverse events as used for other patients. This type of challenge was the primary motivating factor for us for developing PX agent. We have in the past, like last year, developed and published a very exciting model called TXGNN, which was a graph neural network trained on knowledge graph in order to predict drug repurposing predictions. The challenge with that model was it was very hard to personalize it to patient populations. The model was able to take a drug, take a disease, and predict a link between drug and disease, whether it might be likely indication, likely contraindication, etc. The problem is the same drug might be indicated for one patient group, contraindicated for another patient group. These two patient groups have the same disease. And so that is precisely why TX agent supports this open-ended it has an open-ended interface, where in the input prompt you can specify that you are currently interested in patient population that is defined by specific genetic biomarker or specific disease that might have. And so in the context of that patient population, we want to predict adverse events, not just broadly generally. We found that to be much more actionable in terms of downstream validation, which then allow us to faster do experimental validation with organoids, for example, or emulation of clinical trials, because the output of the model that the model provides is more precise, can more directly translate to experimental test. TX agent, regarding the hallucination, so we don't have any theoretical guarantee on hallucination rate of TX agent. Empirically, we have seen its hallucination rates to be much lower than that of other models. And probing the model and studying model behavior, the way this happens is because the model has to ground each step of its internal reasoning in certain knowledge that will be piece of knowledge retrieved via its tools. And so that means that it will not solely rely on the parametric knowledge that is stored in the weights of an LLM, but also will rely on knowledge from external resources. Now, if external resources are fake and all that, that's a different question. But this idea of reasoning, grounding reasoning process in tools is what helps with lower hallucination rates. Okay, if it's okay with you, I will, in the time I have, just make a quick walk you through another vignette that's more molecular oriented, and then I'll take some questions. Okay. So far we talked about therapy. So unit of analysis in all of the examples that I showed to you today were patients. So we have a patient, human, or a group of patients, and we want to reason about them. Well, what if we really do not have patients, but are thinking about these problems at the molecular scale? So we need to think about molecules. So I will share with you some of recent work in this space. And the motivation for the work here was observation that is not hard to make. There's a broad community or a field nowadays. We have models that can predict protein structure from primary amino acid sequence and can do so very accurately for many protein classes. These type of models can achieve atom level accuracy. That was recognized with the Nobel Prize in the last cycle. It's also now not so uncommon to see experimental structural biologies to work with those computational predictions and use them as drop-in replacements of experiments, which is really exciting. There is, however, another grand challenge that is largely open, which is that of how can we understand what are the roles or functions of those molecules? In part, the reason why we want to understand their structure is in order to use that structure to better understand the function. So predicting protein function, the roles, activities that those molecules activate and participate in human cells from sequence or from structure is largely still an open challenge. 20% of human proteins lack well-defined functions. 95% of all publications in life sciences focus on 5,000 most well-studied human proteins. There are 20,000 human protein coding genes, hundreds of thousands of other human proteins and isoforms. Just imagine what we would be able to do if we would understand really what those other proteins that are poorly functionally understood do. So we embarked on this challenge of building a model that can predict function for each and every protein. And so as part of that, we developed a model called Procyon that is a multi-model foundation model for protein function at large or protein phenotypes more broadly for understanding, predicting, and generating those phenotypes. What does that mean? In the simplest case, when the model is trained, human scientists can use and interact with the model in three different ways. In panel A, you can see how the user can specify a task or describe a biological process they would like the model to identify proteins that likely play a role in that biological process. And Procyon will retrieve, score, and rank prioritized proteins against that function. In panel B, one can engage with the model through question answering. So you can ask the model a question about a protein. You can encode in the input prompt not only text, but also information about the structure or sequence of the protein. And the model will say, well, yes, I think that's true, or perhaps I don't think that's not true. And finally, the model has not only predictive, but also generative capabilities where the user can provide in the input prompt information about the protein they want to understand better, and the model will generate free-form text descriptions. Think of them as protein captions or protein cards, which are these phenotypic descriptions about what are the roles and functions of the input protein. So here's the architecture of this model, and this model was developed and his work was led by Owen, who is here and will have a poster, I've heard, in the next session or sometime this week, so make sure you check that out. And Yepeng, Robert, and Valentina, who I think you all know very well as one of the organizers of this summer school. So the architecture is very high level, shown on this slide. It goes from left to right, where you can see that either the retrieval or the generation prompt specified by the user is then decomposed into modality-specific information, where the structure of the protein, if provided in the input, will be processed by a protein structure encoder, which is an atom-level equivalent graph neural network. Amino acid sequence of a protein is processed by protein language model, and the natural text is processed by an LLM that will then either be routed to a text generation arm to generate new text in conditional manner, or in the protein retrieval arm, we have projectors that jointly map the sequence information about the protein, as well as the textual description in the input prompt, in order to then produce a unified text protein latent space from which we can retrieve proteins and prioritize and rank them. How did we train Procyon? To train Procyon, we built a protein instruct training data set, which is a data set of 33 million instructions, where each instruction can be thought of as a tuple consisting of a protein, ID in its associated sequence and structure, and second, information about the specific function or phenotype that that protein might have. And then there is also a protein language encoder that has broad knowledge of evolutionary relationships that are derived from protein sequence, the structure encoder that captures geometric atom-level interactions, and these LLMs that are trained on broad scientific knowledge. So the model was trained to have three main objectives, including next token prediction for language modeling and contrastive learning for accurate retrieval and ranking of proteins against against protein function provided in the input, and finally an instruction tuning objective and ensure that the model better follows human instruction provided in the input prompt. And that led to an 11B parameter Procyon model that is a model whose weights as well as training and inference code are publicly available. Procyon has many exciting capabilities. I will just highlight a few applications in the next few slides. But very briefly, model is multi-model. By that, I mean that it can seamlessly interleave text with structure and sequence of proteins, both in the input and output. The model is capable of doing zero-shot task transfer, meaning it can consider and model tasks and biological functions that it has not encountered during training time. It can do so by modeling functions that do not correspond solely to function labels that exist as predefined functional categories in ontologies or taxonomies, because it has unbounded vocabulary in the user input. And finally, it has both predictive and generative capabilities. So first, benchmarking. On benchmarks, where the model is asked to predict functional categories ranging from disease mechanisms to drug targets to enzymatic numbers and others. The model performs well. It generally performs better than models that are single modality models, such as protein language models alone or structure-based models alone. It also performs better than other recently developed protein text models, and also performs better than more conventional workflows developing computational biology, including canonical BLAST approach for protein function prediction. So seeing that strong benchmarking performance, we were encouraged to test Procyon and apply it for interesting biological applications. I'll show just two of those. So here's one example where we were interested in using Procyon to identify what are peptides, which are short amino acid sequences that likely bind ACE2 enzyme. And so in order to identify which are peptides that likely bind ACE2, we started with a small data set of naturally occurring peptide protein binders and a pre-trained Procyon that we fine-tuned with that small data set to build a version of Procyon called Procyon Bind. And then Procyon Bind was then asked to prioritize and rank peptides that were generated and synthesized experimentally. And after the model predicted those scores of potential binding between peptides and ACE2, we checked, well, whether those predicted scores are informative of actual experimentally measured binding between peptides and ACE2 protein. And so testing the performance of Procyon Bind on this experimental data set, which was generated after the knowledge cutoff date of the model, you can see here in red that peptide ACE binders with higher predicted binding score by Procyon were also enriched in those peptides that have better binding profile to ACE2 determined and tested experimentally than not. Here is now another application, an example, this time generative application of Procyon, where we were interested in using Procyon to predict functions of proteins that are very poorly functionally understood. So AKN81 is a gene whose protein was recently implicated in Parkinson's disease. But when you look at that AKN80 profile in canonical protein functional databases, you can see that the pages of this protein are relatively empty. We don't really know what this protein does much. So we wanted to use Procyon to generate these caption cards or protein images or phenotypes. So going from the left to right, the user provided in the input AKN81 amino acid sequence, its predicted structure, and prompted the model to generate phenotypes that the protein would likely play a role in. And so Procyon then generates various phenotype descriptions, which we can additionally filter to the technique called QA filtering. And so we then checked whether those phenotypes that were generated by the model can be validated through lines of experimental evidence. There are various lines of evidence that we're able to provide for some of the structures, including for the first one, which is a hypothesis about the involvement of AKN81 in citing cytoskeleton, which we can confirm to the protein image to the human protein atlas with images of localization of proteins in different cells. Okay, so if I have just 5 more minutes, then there's the very last thing that I will show. Yeah. Okay, great. 5 more minutes, not more. Okay, so Procyon reasons about one molecule at a time, one protein at a time, and understanding the functions of those proteins, which is already incredibly impactful. But recently with Ada, we were also interested in seeing whether we can start more concretely understanding interactions between molecules. And specifically, that the kind of interactions are those that take place in specific interaction interface between those two molecules, and whether we can start modeling that interaction interface by generating strong representations of interaction interfaces. So that led to a model called Atomica. Atomica is an all-atom model. It's a result of a geometric deep learning processing that's run on atom level representations, different kinds of molecules, and it can generate embeddings or representations on interaction interface, which is the surface of the two molecules where the two molecules come together. So we can now generate embeddings not only of whole proteins, of parts of proteins, but embeddings of the interface between one protein and another protein, or one protein and a small molecule, or one protein and a peptide, and so on. So how does Atomica work? So we have an interaction complex, which is a structure that consists of multiple molecules. We can look at interaction interfaces of size 8 angstroms on those interaction complexes. We represent them as graphs of atoms, edges between those atoms, and then higher order blocks. So that can be common chemical motifs or protein residues or protein nucleotides. And then once we have those interaction interfaces represented, we can train an very fast neural network with a tensor field update to really model well the chemistry of those interactions taking place at molecules. The key advantage of Atomica is because it's an all-atom model, it's not limited to any single interaction type. So the universality of Atomica is that it's a model that's trained across molecular interactions that span also scales. So it's total 2 billion interaction complexes that include small molecules and how they interact with each other, protein peptides, protein DNA, protein RNA interactions, and so on and so on. I want to show that because we recently just completed a very cool experiment with generating this universal Atomica, the latent space, and then Ada had an ingenious idea to see whether there are proteins called dark proteins in this space that we could now functionally annotate and could not functionally annotate them other ways. So she ran Atomica, select a subset of proteins in the dark proteome, and ask Atomica to make predictions based on interaction interfaces. What are the functions that are defined by interaction properties and structure of interaction interfaces that those proteins might have? And for 5 proteins, Atomica predicted that they are likely heme binders. So we then took those predictions, and our friendly collaborators in the molecular lab at the chemistry department, they have ran functional assays for those proteins by partially synthesizing those proteins. We partially had to synthesize them separately, and then run the experiment to confirm that those proteins are actually heme binders. So it looks very cool. You will see the next few slides, not slides, just a second, so they can actually, they have software, they can put in amino acid sequence, so I can generate, run, and now we'll see the robot start this process. It's like very cool. And so that allows us to now do multiple rounds of campaigns. So far we're at round number 1, but we're already in round number 1, 5 of those proteins that Atomica predicted as likely heme binders were experimentally validated. Okay, so with that, I will stop here. It was really a pleasure meeting you all. I want to thank team of students I really am fortunate to be working with at Harvard, our various funders, and happy to take any questions. I will also be here for the next few hours if you want to ask me anything offline. Thank you.